kinetics, a revolutionary tool that neuroscientists use every day for controlling neural activity with light. And he also developed expansion microscopy, which essentially expands tissue in a really creative way and allows nanoscale imaging with standard microscopy. His lab continues to develop many cutting-edge technologies to map, monitor, and repair brain health and brain function, and we will hear a lot of this today. Dr. Boyden has received numerous awards, including the Breakthrough Prize in Life Sciences, and he is an elected member of National Academy of Sciences. So please join me in welcoming Dr. Boyden.
Great, thank you for the invitation to come speak and for the kind introduction. It's a great honor to be here and really excited to tell you about the kinds of things that we're doing in the lab. So I run a group at MIT just across the river where we build technology. And the technologies are driven by scientific questions. We really want to understand how biological systems work in terms of their parts. And of course, the issues of biology compared to other sciences are due to differences of spatial scale, temporal scale, and heterogeneity, right? So in physics or chemistry, you have a small number of things and a small number of ways they interact. But in biology, we have a huge number of things and a huge number of ways they interact. And so today, in the next half an hour or so, I want to tell you about 3 technology sets that we've been developing where we are trying to cross those spatial and temporal scales while not losing sight of the heterogeneity of the building blocks of living things. And so in the first half of the talk, I will talk about a technology for mapping systems down to their fundamental components, expansion microscopy. And in the second half of the talk, I would like to talk about the imaging and controlling of living systems in terms of their signals. So why do we need to map across spatial extents? Well, biomolecules are nanoscale objects, and they interact over nanoscale distances. And everything else, cells, tissues, and organs, are made out of those things. So ideally, we could map a large-scale object in terms of those parts with resolution enough to see their interactions. And there are many ways of doing nanoimaging. Here at Harvard, for example, Li-Huei Tsai pioneered storm super resolution microscopy as just one example of many. But of course, if you want to image a large object, that is still very difficult. And if you want to get down to extremely fine resolution while looking at a large object like a cell or a tissue or an organ, that remained quite difficult when we started the project. So our idea was, what if we take a biological system and infuse it chemically with a dense spider web-like mesh of effectively baby diaper polymer, a swellable material. Do it just right, and I'll show you how in a second. Add water, and you can make the object bigger. And so this started with 2 then grad students, Fei Chen, now a professor at Harvard, and Paul Tilburg, who worked with me on this technology. And so in panel B is a piece of the mouse brain, a couple millimeters on a side, so a fairly large object for a nanoimaging problem. Panel C, about a day and a half later, we've blown it up 100 times in volume, 4.5 times in each direction. And the polymer starts out very dense, like in the upper left in that cartoon, and then at the end of the day, it's stretched out, like in the lower left. Here's a movie, half an hour, sped up to half a minute, to show you it in action. So we formed the polymer earlier, and then we add the water about right there. And so I hope you can see that this piece of the mouse brain is growing as more and more water is added. Okay, so what's happening? Well, all technologies owe a debt to earlier science, of course, and 2 papers that come to mind as paving the way include this wonderful paper in 1980 by Tsuyoshi Tanaka, an MIT physicist. And he worked out the phase transition-like physics of swellable hydrogels. So you can take a hydrogel like sodium polyacrylate, that's the actual swellable polymer we use, and add water. And as shown in this cartoon, what happens is the water is drawn in through osmosis. The polymer threads, shown as white lines, swell apart, and you get this huge volumetric increase. Now, you can't just put the polymer on top, you have to get it inside. Strangely, in 1981, Christian Dreyer and Peter Hausen showed how to do that. They did polyacrylamide hydrogel embedding of cells and tissues, and showed that these preserved embryos and tadpoles and so forth could be stained and imaged better when they were stabilized by this hydrogel. They used polyacrylamide, that's the uncharged cousin of polyacrylate, polyacrylate being negatively charged, like charges repel, that's what causes that huge increase in swelling. But it's fun to think that if somebody connected these two papers, maybe this talk could have been given a half century ago. So the goal then is to take this golden cell on the left of this cartoon and make it look like the cell on the right. So two molecules that we're touching are now some minimum distance apart, and if two molecules are some distance apart, it's scaled up by a linear factor. I made a frequently asked questions slide because sometimes at the end of the lecture, people ask these questions, and by then it's too late. Sometimes people say, oh, have you injured the specimen? Is it unhealthy? It's completely dead. This expansion process is not compatible with life. Let's just get that out of the way. Sometimes people ask me, oh, have you disrupted the molecular interactions? Yes, they're all gone. We pulled them apart from each other. But the information is still there, right? Information in biology, in large part, is encoded by the relative organization of molecules, right? Molecules work by touching, and when molecules touch, then you can cut or bind or change shape. And so if we can map out the molecules and how they're organized, that's often at the core of many scientific questions that we're trying to answer. Okay, back to the regular talk. So these cartoons show you how it works. We had to invent 4 kinds of chemistry to make it work. The first is anchoring. So in this cartoon, biomolecules like proteins are shown in brown, and in purple are little handles that we attach through covalent bonds. And we now have handles that can bind to proteins, nucleic acids, sugars, lipids, and the list increases all the time. Step 2, form the polymer. We use free radical polymerization to weave that polymeric mesh in between and around biomolecules. So we really want to make the mesh of the polymer very, very dense, right? That's how we make it precise. And importantly, when the polymer chain encounters a handle, then you form a bond. So now the polymer can swell, the handle can convey the force. We're almost there. But there's a third step, and this is one of the trickiest ones, softening. We can use enzymes, heat, detergent, or all 3, and loosen up the molecules from each other. Or sometimes we just destroy molecules that we don't need anymore. Then, when we add water, the sodium polyacrylate, the baby diaper polymer, will swell. But because of the softening, because of the polymer, and because of the anchoring, we're pulling things apart from each other. And also, you can see the topology. It's cross-linked so that the polymer threads expand evenly. We don't want them just to pull apart from each other randomly. So by design, the expansion process was intended to be as even as possible, but this is biology, we should show it. And so we spent a lot of time, especially in the early days, doing pre-expansion imaging with a classical super-resolution method, like STORM or SIM or others, or DNA Paint, also developed here at Harvard by Peng Yin. And then we do post-expansion imaging with a regular old classical microscope. And so here in pink is pre-expansion. This happens to be SIM microscopy. In green is post-expansion, and then we overlay it. And the arrows show the amount of error that's being introduced by expansion. It's not 0, but it's really, really small. It's like a couple % over a microscope's field of view. And so compared to like microscope and fixation and other errors, it's very, very, very negligible for the most scientific questions. That's distortion. What about resolution? Resolution is defined as how close two things are that you can tell apart. And so we know what microtubules, such as in this picture, look like. We know how viruses look, all sorts of sort of natural scale bars, if you will. And we can then gauge the resolution. And so with 4.5-fold expansion, we're getting a resolution of around 70 nanometers. Now, a typical good classical microscope might have a resolution of 300 nanometers. 300 divided by 4.5 is about 70. And so you might wonder, if you expand more, can you get better resolution? You can, but with 4.5-fold expansion, there are a lot of things you can do that are really hard to do, and sometimes even impossible to do with earlier techniques. And so the first thing we want to do is to image a large chunk of brain tissue, which is very hard to do with older methods. So on the left, I'll use the mouse pointer. We're zooming in. Each white square is zoomed in, the picture below. The color code shows how we're labeling different proteins with fluorescent antibodies, because that's how light microscopy is often done. The left is pre-expansion, and the right is the same specimen, same microscope, but after expansion. And I hope you can see the image is much more clear on the right. In fact, the purplish fuzz in the lower left can be resolved into those clean pink and blue subdomains. And those are pre and post-synaptic proteins, bassoon and Homer. And if we look at the distance between those protein densities, we get the same answer that Catherine Dulac and Jiahui Zhuang measured many years ago with STORM super resolution. But now you can make that measurement on a conventional classical microscope. Eric Betzig, another super resolution pioneer, applied his light sheet imaging method to our expanded objects. When you expand something 100 times in water, it's mostly water. And so you can image by shining light from one angle and taking pictures at a 90-degree angle, and you can go very fast because every camera frame is taking a whole 2D slice of your image. So you can go blazingly fast, much faster than older methods. Here in the upper right, we're looking at mitochondrial and lysosomal proteins, again, antibody stained. At the bottom, myelination, work spearheaded by Reigao Sho Asano and Gokul Opadiula. Those are all natural proteins, but you can express artificial proteins, like fluorescent proteins that are mutants of jellyfish and coral proteins. Here we're expressing using the rainbow technique developed by Jeff Lichtman and colleagues here. So every neuron gets a barcode, if you will, a combination of fluorescent proteins. And so we're in the mouse hippocampus here. This white square, we zoom in. It's sort of hard to see the wiring of the brain because it's nanoscale, but after expansion, you can cleanly resolve the fuzziness into individual neural wires. And so many, many groups now are using some combination of a stain and expansion to map brain circuits. That's all protein staining. What about other biomolecules? So when Oswasi joined our team, and later Gaoqi Yang and Li-Huei made a very inexpensive version of this, we anchored and expanded apart both proteins and nucleic acids. So here you can see yellow fluorescent protein in green, and each pink dot is a single messenger RNA that we're doing post-expansion labeling with fluorescent in situ hybridization. So in intact brain tissue, you can look at the location with nanoscale precision of individual molecules, in this case, mRNA molecules, and map out how gene expression is spatially regulated with great precision. That's all about fourfold expansion. What about bigger expansions? Well, you can change the recipe and get bigger expansions, but eventually, of course, it runs out of steam. You eventually would fall apart. And so when JB Chang was at our group, and later Deblina Sarkar, Jin-Young Kang, and Oswasi developed a really easy version of this, we found a way to iterate expansion. Now, what does that mean? Take a specimen, polymerize it, and expand it, form a second polymer in the space opened up by the first expansion, and expand it again. And you can do this over and over again. This gives you any resolution in theory that you want, but it illustrated in this cartoon is another advantage of expanding something, decrowding. Now, in light microscopy, we're visualizing fluorophores attached to biomolecules. So in this cartoon, the red and green antibodies can decorate proteins on the outside of a complex, but they can't get inside. But if you expand the proteins away from each other first, then the yellow and blue antibodies can make their way in. Synapses are jam-packed full of proteins. So we wondered, can we start to image the relative organization of these through post-expansion staining? Working with Li-Huei Tsai at MIT, who you'll hear from later, and also Tom Blanpede at the University of Maryland, we started looking at dense protein clusters, such as amyloid, and also proteins at synapses, and found that a large variety of proteins we could stain better after expansion than before. In the interest of time, I'll show you one set of images here. In yellow is pre-expansion staining, and there's not much yellow. And then pink is post-expansion staining. It's a crowded slide, but this is a calcium channel, RIM12, PSE95. And in all of these cases, you get better staining afterwards than before. Not all proteins are crowded. Here in F, G, and H, we have Homer, bassoon, Shank3, and the yellow and pink, I hope you can see, look very similar to each other. So you can do things like look at the nanoarchitecture of synapses, of amyloid, of all sorts of interesting things, and map out the relative organization of biomolecules with respect to each other. I should point out, Jin-Young Kang and Margaret Schroeder went on to show that there's another nice feature of decrowding. You can stain, image, and wash over many, many cycles. And so this just shows a schematic of the protocol, but it's all published. So I think I'll just show you the data. Here we're looking at 20 different proteins with 20 nanometer resolution in intact brain tissue. So they're staining about 5 antibodies at a time. And these are all the same field of view in the upper left. So there's 4 images in the same field of view. And the dotted boxes are zoomed into over here. These are individual synapses. And you can start to see, almost 24 proteins and how their relative organization is at synapses. And so these data sets are beautiful. I mean, you can just sort of see all sorts of patterns by looking at them. I should point out, in a collaboration with Laura Kiesling's group in MIT Chemistry, we have shown that we can do the 20-fold expansion that you get through iteration. 4 times 4 is about 20. But you can also do it in a single step by some clever tricks, like optimizing the oxygen and so forth. Work led by Tei Xin and Shuwei Wang. Paulo Valdez, a neurosurgeon working with us and Nino Kyoka at the Brigham and Women's Hospital, wondered if we could see invisible proteins, could we see invisible cells? Now, aggressive tumor cells have high levels of vimentin, shown in cyan, and GFAP, shown in pink. These are actually human specimens from brain cancer patients. And the first couple rows with pre-expansion staining. The fourth row is the same specimen, same antibodies, but with the antibodies reintroduced a second time after decrowding and expansion. And Pablo convened a team to count the number of putative aggressive tumor cells, these vimentin GFAP double positives. And with post-expansion staining, they counted 6 times more of these tumor cells than before. So we're very excited about the possibilities that this could help us characterize lots of different pathology outcomes. Yongshin Zhao and Octavia Bucher, working with me and with a whole set of Harvard pathology faculty, Astrid Wines, Stu Schnitt, Andy Beck, and others, showed that we could adapt expansion to non-brain tissue types, so prostate, lung, breast, pancreas. On the left are normal, and on the right are cancer-containing specimens. And I hope you can see that the post-expansion images have better resolution than the pre-expansion ones. This slide really isn't meant to be read, but the number of groups doing expansion grows every day. I think the number is in the couple thousand by now. The number of experimental papers, not like review papers, is about to hit, I think it actually crossed 800. This slide is out of date. And then our protocols are at expansionmicroscopy.org. Nature did a little 10-year anniversary news story about expansion. And one of my favorite quotes is from Yongshin, now running his own lab at Carnegie Mellon. And he says, expansion is so easy, even my 9-year-old kid can do it. So we posted our protocols on the web, including photographic tutorials. We're about to start filming some video tutorials as well. So please check them out. And if you have any questions, I'm happy to answer them. Now, expansion is interesting because it's not only easy to apply, it's easy to change. And so this is an unexpected plot twist, but many groups are adapting expansion, so often in collaboration with us, for other purposes. So Silvio Rizzoli's group had an interesting idea. We were expanding proteins away from each other. They wondered, could you expand a protein itself? Take a protein, anchor it to the polymer, chop the protein into pieces, maybe using an enzyme, and pull the pieces apart. What does it look like? Turns out it looks a lot like the protein's ground truth structure. So in gray are the ground truth structures with cryo-EM or X-ray crystallography or so forth. And the false color images are single, unaveraged expansions of proteins. So forget about copy number or abundance and so forth. We can look at individual molecules. And our hope is that we start to map out how individual molecules are organized and how they communicate with each other. Another example, this was published in Nature last week from Johann Danzel's group in ISC Austria. They found that nonspecific protein staining plus expansion can give very, very traceable connectomes. You can start to trace with machine learning the dense wiring of, in this case, the mammalian brain. And just one last example from JB Chang, now running his own group at KAIST in Korea. They added a softening step to soften bone, and they can expand the whole body. This is an embryonic mouse. Looks transparent because it's been expanded. You can hardly see it, but that's the whole body, cartilage, bone, and all. So it's very exciting to think that expansion is not only democratizing nanoimaging for everybody, but also anybody could tweak it or tune it for whatever purposes they want. In the last half of the talk, I want to talk about the imaging and control of living things. I'll just briefly mention optogenetics because it's a fairly mature field. But in optogenetics, we want to control brain cell electrical activity with light. This started when Carl Dijsselroth and I were both students at Stanford, and we were brainstorming, just going through all the laws of physics. There's only so many kinds of energy you can deliver to the brain. Which ones could you use for brain control? There's light, there's sound, there's X-rays. There's only a handful of things, right? Then we did mathematical calculations. Light seemed like it'd be the best because you could aim it very, very precisely. You could bring light into the brain. Optical fibers could be brought into the brain, the same way electrodes are brought into the brain, even of awake human patients undergoing neurosurgery. Then the question is how to make brain cells sense light. And so I became really obsessed with microbial rhodopsins, 7 transmembrane proteins that look like the lower left. they sit in the membranes of single-celled microbes and convert solar energy into chemical energy. In the first case, it was a light-driven proton pump that would store sunlight in proton gradients. And people later found light-driven chloride pumps, light-driven ion channels, and more. Flash forward to the current day, what we found, and our colleagues have found, is that members of all of these can be identified that are safe enough, fast enough, and powerful enough to work in delicate neuronal environments. So when Brian Chow and Xue Han worked with me, we found that certain light-driven proton pumps could be brought into brain cells through genetic delivery. The brain cells express the proteins, and you could shine light of the right color, green or yellow, for example, and pump protons out of neurons to shut them down. Light-driven chloride pumps, Xue and later Amy Chuang, found that we could also do neural silencing this way. So by deleting neural activity, you can find out what neurons are needed for. Working with Carl, I found that we could use light-activated cation channels to activate neurons, in this case, in response to blue light. And later, Nathan Klepetke and others worked to extend the toolbox. And so just like expansion being used by thousands of groups, optogenetics, as we call it, opto for light, and genetics, because these are genetically encoded things, are also being used by thousands of groups. And I just wanted to mention one thing, because for the most part, people are using these molecules in animal models like mice, where people are making amazing discoveries. But also people have started a couple summers ago using these in the human body. So a brave team in Europe, led by Bolton Ruska and José Alain Sahel, took one of our molecules, the crimson molecule that Nathan had published in 2014, delivered the gene into the eye of a blind man. And now there are about 30 people I've heard who have been successfully treated with these light-activated proteins from our group. And he has retinitis pigmentosa, so he lost the photoreceptors of his eye. But by delivering the molecule to the spared cells of the eye, he was able to have partial restoration of functional vision. He could see household objects like cups. He could see doors on a hallway. Not perfect vision. He struggled to read text. He struggled to recognize faces. But the people in the field have ideas of how to improve the fidelity of the vision. So I guess with optogenetics, and I want to talk more in the last couple of minutes about newer things that we're working on, really just we were very, very lucky, right? These molecules might have been toxic. Maybe they would have been too slow or too weak. They need an all-trans-retinal, a vitamin A variant to work. That didn't have to be in the brain, right? It's not in the nervous system of, for example, the worm C. elegans. But for some reason that we don't fully know why, in the mouse body and in the human body, there's enough all-trans-retinal around to make these molecules sense light. So it's really just a serendipity story from beginning to end. And so in the last part of the talk, I want to talk about, in the last few minutes, about the imaging of signals, or the opposite of optogenetics, if you will. And in this case, we haven't been quite so lucky in the sense that nature hasn't just given us stuff right out of nature that we can use, but great bioengineers have been able to engineer fluorescent reporters of live signals. So take the green fluorescent protein from jellyfish or a red fluorescent protein from coral, and then you can connect it to a molecule that will bind to calcium or protons or whatnot, and then you can express the gene for this genetically encoded fluorophore in a cell, and you can monitor signals in a living cell. But of course, cells are complicated. I think we've all seen cartoons like this. I just stole this from Wikipedia, but cells take in countless inputs, right? Hormones and nutrients and neuromodulators and transmitters. Inside the cell is this dense network of computation, right? Each oval is a different biomolecule, each arrow a different kind of interaction. And then a cell will make a decision. Maybe it lives or dies. Maybe it moves. Each arrow was probably a different PhD thesis, right? And maybe they were done in different species. Like this one was figured out in a fruit fly, and this one was done in a worm. And so the dirty secret is that these cartoons, we don't really know what they look like, right? In a given cell, in a given patient, who knows what the network actually is? So we started wondering, could we just image the whole thing? As I mentioned, there are lots of fluorescent reporters of signals out there. This is from Jin Zhang's wonderful database from her group at UCSD, where people made fluorescent reporters of different things by connecting a fluorescent protein and a binding domain. And so we started wondering, could we use them together? And basically, to image many things at once in a living cell, we can borrow ideas from, well, basically how the internet works. And so, here in this building, there are lots of different Wi-Fi antennas. If you go from room to room, you can go from one antenna to the next. And so we wondered, what if we could do the same thing in biology? Have different places in a living cell tell you different stories. The other idea is temporal multiplexing. So if we all made a call on our cell phones right now in this room, it actually might work. And it's because our phones have orthogonal codes. And even if we're all fighting over the same antenna, the orthogonality lets us sort out the calls from each other. So we asked the question, what if we could have different temporal codes associated with different signals? So in the interest of time, I'll just tell you one of these stories because we're almost out of time. Shannon Johnson and Chengyang Lingu, when they were in my group, worked out spatial multiplexing. And so this cartoon shows the idea, what if different places in a cell could tell you different stories? So what if the points labeled 1 could have a calcium indicator, and the points labeled 2 could have a protein kinase A indicator, for example? So while the cell's alive, each dot will blink out a little movie of what's happening there. And then when you're done, you can preserve the cell and stain it using techniques like I told you at the beginning of the talk, and maybe over many rounds of staining and washing, right? And figure out which sensor was where. And so it works. Here's the design that we came up with. Take different fluorescent reporters and fuse them to different self-assembling peptides. So they'll cluster stably and safely at different places in the cell, random places, but stable places. And then each reporter has an epitope that you can stain for to figure out which sensor was where. So here is a hippocampal neuron in culture, 3 different reporters, calcium, cyclic AMP, and protein kinase A, fused to different self-assembling peptides. And all 3 reporters, they're all green, by the way. So the green image is what you see when the cell's alive. Each dot will be blinking at you. And the false color images are what you get after you're done, preserve the cell, and stain it. And so you can then reconstruct which signal came from each dot. Here we're imaging 3 signals at the same time in a living neuron while we give it a drug. And we can start to see relationships between the different parts of the signal. So we get this drug forskolin that drives cyclic AMP, and we see that the speed of calcium basically correlates with the amplitude of the protein kinase A. So you can rate the speed of one signal and the amplitude of a second signal. This would be very hard to do with older techniques, right? Because, watching one signal in one cell and a second signal in a second cell, you'll miss out on the relationship between them. Anyway, so you can add a 4th indicator if you want. Here we added a protein kinase C indicator, or a 5th indicator. Here we add an ERK kinase indicator. And so our hope is that we can use this to image a wide variety of different signals in living systems. So I think in the interest of time, rather than introduce a whole new story right now, because I think we're very close to ending time and I want to leave time for questions, I think I can end here. I think I acknowledge along the way all the people who've led those projects, but an even longer list of people have helped over the years. In the middle of the slide, for example, are just the PIs of groups we've collaborated with over the years. We share all of our tools freely, so please visit our website, synthneuro.org. And if you have any questions, my email is just edboyden@mit.edu, and I'd be happy to take some questions now.
It's an amazing list of people. All right, we have time for at least 2 questions.
To allow multiplex imaging, antibodies are very useful tools, of course. But so the expansion microscopy grade antibodies, are they any different from other, IF antibodies, conventional IF antibodies? So validation. So the ones work for IF, are they more or less guaranteed to work on expansion microscopy or not? Because of the tissue processing, so on, is somewhat different.
Yeah, great question. So you can add antibodies at 2 steps. You can do at the beginning, which is when people normally would stain. And so there, the antibodies will have very predictable behavior. So they'll just work. You can also add antibodies at the end, which is what I suggested in this slide. In this case, you get different results. So 80% of antibodies work as well or even better. As I showed, sometimes you get new signals where you did not see signals before. Not all antibodies, though, work perfectly. Maybe 20% will be corrupted. What's going on? Well, we're denaturing and pulling apart proteins. So Western blotting antibodies, antibodies that look at local parts of protein, they'll work well. But any proteins that depended upon tertiary structure, anything about sort of the exact binding partners or conformations or whatnot, those antibodies might not do so well. Sure, that'd be great. Yeah. Yeah, we love collaborating with people, and yeah, I always want to help everybody. And so yeah, drop me a line, and I'd love to chat more.
All right, I guess I will ask the next question. So, this just strikes me. I have known about this expansion microscopy for almost, 7, 8 years now, but it just struck me. Can you use it to like detect some of the physiological changes? For example, during concussion, some axon will be injured in a way that's like not completely broken. And if you expand them, I wonder if people have done that kind of experiment to visualize the differences.
Yeah, many people are applying expansion microscopy to neuropathology,
Okay.
and looking at everything from epileptic seizure, tissues from human patients, from animal models. With Li-Huei Tsai, we have a joint student who's looking at some of the axon degenerations that occur in neuropathology, and we're seeing very interesting things. So yeah, it does work really well on brain. It works very well on lots of things, but yeah, looking at neuropathology and mapping out what happens at the nanoscale is turning out to be extremely interesting. And yeah, many groups have also been applied to Parkinson's disease. There have been a couple papers now on alpha-synuclein mapping. Yeah, it's so far I can still track every paper that does expansion, but it's getting to the point where I'm losing the ability to do that. But yeah, there's a lot of expansion communities as well. Like there's a whole malaria expansion community where they've worked out all the tricks for getting it to work really well with malaria. And there's a whole centriole community looking at like cell division and all that. And they've published like dozens and dozens of papers mapping out like the nanoarchitecture of centrioles. So I think what happens is that different communities adopt expansion and they start making discoveries, then the communities can grow.
Thank you. Sure.
Yeah, so basically all the expansion protocols that we present to the world anyway have been optimized where we want the polymer density to be as dense as possible, and we want the softening to be as strong as possible. So at the beginning, we wouldn't publish a paper until we would see that different things expanded evenly. We would look at white matter, we would look at gray matter, we would look at the nucleus, and if things were not expanding isotropically, then we wouldn't stop. We would continue to improve the softening steps, increasing proteases, lengthening the duration, increasing the temperature, and so forth. So by the time we present a protocol, basically we've maxed out the softening steps. At this point, I think for soft tissues, you can find protocols that will work for a wide variety. So for Yongshin's protocol, for example, and now he's running his own group at Carnegie Mellon and had this beautiful protocol called Magnify, which also works on many different tissue types. Basically, what he's been doing has been maxing out each step in terms of density and softening until you can expand all these different tissue types. And if anything doesn't work, then he keeps increasing the strength of the softening. So he had this beautiful Nature Biotech paper on Magnify about 1.5 years ago, I guess. And that protocol works on all sorts of tissues.
All right, one last question.
Can you hear me? Okay.
Yeah.
Thank you so much for a nice talk. I got a simple question for the last part you mentioned. So for the visualize the cell, my question is, what is the bandwidth now? Like how many targets you can visualize simultaneously? And what is the major limitation here?
For the live imaging?
Yeah, for the last one. Yeah.
Yeah, great question. Let's see. Yeah, so I showed 3, 4, and 5 things being imaged at the same time. And there's one question that one question that I think we're having to wrestle with is just how to deliver many, many genes to a cell. And so, yeah, so one thing we're trying to do is just figure out, to deliver 10 genes to a cell reliably, 20 genes to a cell reliably, how to go about that. And so, we're kind of going into some areas of bioengineering that are not our original bread and butter, but we're trying to ask the right questions there. But I think that's really the key, is just getting lots and lots of these genes in, or some vector that can have multiple genes in it that will express in a balanced way. I think we're close to figuring that out, though. It's just a bit of a detour for us because that wasn't our original expertise.
Thank you so much.
But I would love to be able, I don't know, maybe a good 5-year goal is, can we image 100 things in a living cell? I don't know if we can achieve that, but that's kind of internally how I like to think about the path.
Yeah, ideal situation, we can map everything, like 20,000 genes together. Thank you so much.
Yeah. Yeah, well, there might be other tricks we could develop. Yeah, this isn't the only project we're working on in the space, but yeah, there could be a whole set of tricks that we could build for this, I guess.
All right, let's thank Dr. Boyden again.
Thank you. Thank you so much. Okay.
All right, that was fantastic. Our next speaker is Dr. Isaac Chu, Professor of Immunology at Harvard Medical School. I heard my echo, but all right. Dr. Chu's lab studies the crosstalk between nervous system and the immune system. Particularly, he studies how sensory neurons not only detect nauseous signals, but also interact with immune cells and confer responses. His discoveries on neuroimmune communication have important implications for pain, inflammation, and host defenses. His work has been recognized by awards such as NIH Director's New Innovator Award and the Chan Jacques Ben Barras Award. Today, I believe he will share insights into how sensory neurons modulate immunity at barrier sites like the gut and lung. Please welcome Dr. Chu.
Thank you so much to the organizers, and you know, as somebody growing up here in the U.S. but being Chinese American, I'm honored to be here, and I think it's important how I mean much contributions Chinese scientists have made to the work here. So I encourage you all to keep doing what you're doing, and hopefully things will improve. So it was hard to follow Ed, but I thought that was amazing, and maybe I can contact him afterwards about some of the techniques he's doing. So we're really interested in understanding crosstalk between the nervous system and the immune system, which we think have developed ways to communicate, because it's important that these systems coordinate both physiology and also host defense. So pain is one of these systems where we think there's parallels. So, you know, pain is actually one of the 4 cardinal signs of inflammation, as defined by Celsus, who's the, 2000 years ago, the Roman physician. Dolor, right? So dolor is pain, there's redness, heat, swelling. So in a sense, immune responses and pain have long been linked together. But we're starting to appreciate that the nervous system that mediates pain also coordinates inflammation itself. And so these neurons that mediate pain are called nociceptors. They're the ones that detect noxious stimuli. They innervate especially densely barrier sites, so where we might get something injurious or harmful, like the skin, the lungs, the gut. And they're sensing signals from the environment, but also internally that might be potentially harmful. And these include things like noxious temperatures, extreme heat or cold, also potentially damaging chemicals that we might be exposed to, as well as tissue injury or force or release of damage-associated molecules like ATP, and also pathogens, which is another area which I think there's interesting parallels, because when you get infected, you get sick, and you get pain as one of your main signs of infection as well. So how do these neurons sense noxious stimuli? Well, one mechanism is through ion channels, ligand-gated ion channels, such as TRPV1, which was discovered by David Julius, who won a Nobel Prize for discovery. So this particular ion channel is gated by, he discovered capsaicin, which is that active ingredient in chili peppers that causes that spiciness, right, or pain. But of course, it's evolved actually to detect noxious heat. So it's opened when it senses temperatures greater than 45 degrees and also low pH. So this is expressed by a large proportion of our nociceptive sensory neurons. And I'll talk, I'm bringing it up because we also use it as a way of targeting nociceptors to see how they play a role in specific immune responses. Our lab and others have also over the years found that these neurons can sense pathogens directly. So like immune cells, they have different ways of sensing potentially harmful pathogens. So we found that Staph aureus and Strep pyogenes, two leading causes of skin infections, actually causes pain by directly poking holes in the membrane of nociceptors through these pore-forming toxins like streptolysin S or alpha hemolysin. There are also parallels with innate immune cells where some of these neurons express toll-like receptors, TLR4, TLR5, which can detect gram-negative bacterial cell wall as well as flagellin. And then fungal pathogens also cause pain, and viral pathogens like herpes virus can sensitize these neurons, right? So in the setting of infection, they're not only, sensing potentially inflammatory mediators, but also pathogens. And in response, then they can coordinate host defense. So they can, in fact, also talk to the immune system. And so I'll get into two mechanisms by which that happens. So they can sense pathogens and coordinate immunity. The first one is directly talking to immune cells. So even though these are sensory neurons that transduce signals to the brain to mediate pain, locally when they're activated, they also release neuropeptides from their terminals called, so for example, CGRP and substance P. And immune cells actually have receptors for these. So immune cells harbor receptors. Macrophages, for example, can sense CGRP, and this modulates their cytokine production. And mast cells, which are kind of immune cells that have all these granules that are stored there, they can sense substance P, and this will induce inflammation. So you can get immediate coupling of pain with the immune response through this local, I would say, efferent function. And another key way is through brain-body reflexes. So you have a pain signal going up to the brain, and then this is coordinated to induce sympathetic or parasympathetic reflexes through autonomic nervous system back to the body. And these brain-body connections then modulate the immune response in the periphery. So we have long distance as well as local interactions where nociceptors can coordinate immune responses. So I'm going to tell you a couple of places where we think this is important. So one is in the gut, and this is work that was in collaboration with Christophe Benoit in my department. So Kim Mearshart in my lab and Yang Zhu in the Benoit and also Diane Mathis's joint lab did this work. And so we were just interested, actually starting off not specifically in pain, but trying to define neuroimmune crosstalk in the gut, which is one of the most complex organ systems where there's lots of different nervous interactions. And there's this gut-brain axis where you have signals going from the gut to the brain and vice versa. So we want to know if we activate different types of neurons that innervate the gut, what happens to the immune system? So we use what's called chemogenetics, which is a tool that where we can target different types of neurons using different Cre lines. There's 8 different Cre lines here. TRPV1 is one of them, which marks nociceptors. And what we can do is we can put in these DREADs, which are designer receptors that can respond to a specific synthetic ligand, CNO. So what we did is we targeted these DREADs into each of these subtypes of neurons, which you can see is labeled here in red, and then we activated them using this CNO. And then we just said, okay, what happens after 14 days of activation of different subsets of neurons? Some are enteric neurons, so you can see they reside in the enteric nervous system. This is the gut enteric nervous system. Some reside in the nodose ganglia, which is in the vagus nerve that innervates the gut, and the DRGs, which mediate pain. And so we saw a lot of different results, of which there's kind of a matrix here, which I won't get into details, but basically the point is that depending on the place in the gut we're looking and the type of neuron we activate, there are different changes in different immune populations. So the specific one that we're really interested in is the TRPV1 neurons because of nociception. And they had the largest effects. They actually caused changes mostly suppression of different immune cell types in the gut, of which one that's quite interesting are the regulatory T cells. So Tregs are really important to control inflammation, right? So T regulatory cells produce IL-10 and anti-inflammatory mechanisms to dampen things like autoimmunity or to induce oral tolerance. And you can see here when we activate the TRPV1 neurons using these DREAD approaches, over the course of time, you start seeing Tregs going down, in particular this subset that expresses RO gamma. So what does this mean? This means potentially pain will induce the loss of the control of inflammation. And how does this work? Well, it turns out we found that these neurons signal to the Tregs via this neuropeptide I mentioned earlier called CGRP. CGRP signals via its receptor called RAMP1. So if we remove RAMP1 conditionally just in the Tregs using FOXP3-CRE, this neuronal suppression of Tregs goes away. And you could see actually localization of CGRP positive nociceptors in the gut next to Tregs. So this could be cool to look at by expansion microscopy, by the way. I was just thinking as Ed was talking about. So I think that there are nerves that are next to immune cells and they can talk to them through, in this case, a neuropeptide. We also showed pharmacologically, if we block this receptor for CGRP, which is formed by RAMP1 with its partner, CalCAR-L, which is a G protein coupled receptor, this also inhibits the ability of nociceptors, TRPV1 neurons, to suppress Tregs. Just as a note, this particular drug or receptor is a target for migraine. So people who have migraine often take this RAMP1 antagonist, which the number one side effect is actually gut-related things. So there could be something also happening with T cells in the gut. But what we see is if we did this activation of these nociceptors and then challenge them afterwards with a DSS-induced colitis, so you need things like Tregs to control inflammation. So now you get worse off outcome of colitis, where you have, in this case, more weight loss and also greater pathology and inflammation. So just a brief summary of this first part. In the gut is one area where we think there's a complex neural immune interactions. There are lots of different subtypes of neurons, of which one are these pain fibers called TRPV1 nociceptors. And they seem to be, when you activate them, it actually downregulates Tregs, which increases inflammation. So the pain and Treg systems are actually counterbalanced. There was a recent science paper showing that Tregs actually block pain. So it's possible that these mechanisms are important counterbalances in inflammation and neural responses. So I'm going to now switch gears to a second organ where we think these nociceptors or sensory neurons control inflammation and really balance that, and that's the lungs, the respiratory tract. So in the airways, you don't necessarily feel pain, but there are sensory neurons that are analogous to the pain-mediating nociceptors that reside in the vagus nerve. So the vagus nerve, which this is actually an image from Steve Liberlith's lab, who we collaborate closely with, innervates all of the visceral organs, including the lung, the gut, the stomach, and the heart. And there are very diverse types of neurons in the vagal ganglia, including TRPV1 expressing neurons. And so if we inject an anterograde tracer virus into the vagal ganglia with the TRPV1-CRE, you can see nice innervation of the airways. So what are these nociceptive neurons in the lungs do? Well, one of the things they do is mediate a protective reflex, which is cough. So you can see these are actually TRPV1 neurons innervating both the trachea and the alveoli. And if you imagine, if you breathe in capsaicin, which actually people use for pepper spray, right? Or tear gas, that's triggering this cough reflex because these neurons then trigger a sensory motor reflex. So we want to know, could these neurons also interact with the immune system in the context of inflammation? And we specifically were thinking about infections like viral and bacterial infections. So flu virus, as you know, is one of the leading causes of acute lung injury and death. And somewhat, it depends on the pathogen, how you end up dying. So for flu, one of the key causes of death is actually from immunopathology. So this is a 1918 pandemic lung. You could see this huge accumulation of immune cells and also fluid. And then Paul Thomas, this is just a review from him showing the cytokine storm, that these uncontrolled inflammatory reactions also drive death. So we want to know, what if we target these neurons, TRPV1 vagal neurons, in the context of infection? So this actually started a few years ago where Pankaj Baral, who's now an assistant professor at Kansas State University, tried to first look at a bacterial infection model. So we took Staph aureus, which is a cause of lethal bacterial pneumonia, and we treated mice to deplete them of these TRPV1 neurons in two different ways. One is using a TRPV1 DTR mouse, where we can kill off the neurons with diphtheria toxin receptor injection. And then if we gave them Staph aureus afterwards, you could see they actually did much better. They survived more than the control, and they maintained better body temperature, and they cleared the bacteria more. Here's a second method where we give the mice this high affinity TRPV1 agonist called restifrotoxin. It's from a cactus that's extremely painful. It's 1,000 times more painful than capsaicin. But because it's such a strong stimulus, it will kill the nerves. So we ablate chemically the TRPV1 neurons and then give them the Staph aureus. And you can see, again, they did better. They lived, had higher mortality, less mortality, higher body temperature, and also better clearance of the bacteria. So what's going on here? So it turns out one of the things that happens is if you lack these neurons, either in the RTX or TRPV1 DTR mice, you get faster recruitment of neutrophils. And neutrophils are really important for clearing bacteria. So early on, they actually rush in more. So the neurons seem to be suppressing neutrophils. But this is counterintuitive. You're detecting a bacteria, maybe harmful, and actually this is bad for controlling it. So this led us to ask, what happens if we give it a virus? And I mentioned influenza virus. So Pankaj then basically infected these, again, the same exact animals, knocking out TRPV1 neurons with flu virus, and then looked at both immune response and antiviral and immunopathology. And so here he's giving this, again, this chemical ablation approach, RTX for his vehicle, give them PR8 influenza virus, H1N1, and it's the reverse of the bacterial infection. So here, RTX treated mice did a lot worse. They survive, they have worse mortality, and also there seems to be potentially a sex difference. So females do worse than males. So this is two different bugs, the same exact mouse, but opposite phenotypes. So what about the vagal neuron? So here, we specifically ablated vagal sensory neurons that express TRPV1 by using this TRPV1 DTR mice, where we just injected DT into the vagal ganglia versus PBS. And then when we gave the PR8 influenza virus, they also did a lot worse, you can see, than the PBS control. So what about if we try to activate the neurons? So Pankaj actually gave the mice capsaicin, which is the chili pepper compound, prior to infection. And again, we see this sex difference, but this actually protects the mice. So in males, but not females, from the lethal outcome of infection. So it seems like TRPV1 neuron activation is good and ablation is bad in flu infection. So what about, the first question is, does this affect the viral clearance, right? So we could measure that by, in this case, measuring the levels of nuclear protein in the lungs. And over the course of infection, there seems to be similar levels of the virus, at least by transcript in the lungs. So it's not overall viral clearance, but actually Nicole in my lab, who recently did this immunostudy, did see there was a big difference in viral spread, even though the overall amount wasn't different. If she stained for NP protein, what you could see is in the lobes, there's a lot more viral spread. So the neurons seem to be important in controlling or restricting the virus from spreading too much in the lungs. So this is then accompanied by something else, which, as I mentioned earlier, is really important for actually causing death in humans, is immunopathology. So if we looked at histology by H&E, you could see that in the mice lacking the nociceptors, RTX treated mice, there's a lot more fluid accumulation, especially at these later time points, plus influx of immune cells. So we thought maybe that's what the role of the neurons are, is to control the immune response. And so what we did is we looked in the bronchiolar lavage fluid, and actually, Pankaj, this is at day 7 post-infection, you could see that in the lack of nociceptor mice, there's also this bloody accumulation in the RTX mice. And then we did a cytokine multiplex array, and several cytokines, which normally are lower, are now much higher. So here's just some examples. So I think this kind of cytokine storm I mentioned is exacerbated. So it's many different things. IL-6, CXCL1, which is actually a key neutrophil chemokine, goes up, and GCSF, which also stimulates neutrophils and myeloid cells, but also things like IL-13 and IL-17, which are characteristic of other types of inflammation. So seems like just uncontrolled immune response and also cytokine production. So to dive deeper into what's happening in the immune system, we did single-cell RNA-seq of the lungs in both RTX versus vehicle mice. And what we found was the most striking differences were in the myeloid population. So the neutrophils and actually monocyte-derived macrophages, those go up significantly in the absence of these neurons, whereas other like lymphocytes actually went down. So if we also look at the gene expression profiles of these innate immune cells, what they're doing is they seem to be dampening the antiviral immune response. So you have less type 1 interferon responses, or ISGs, which are really important for fighting things like flu. And then you get upregulation of things like in the IL-1 signaling pathway. So the polarization of these cells seems to be also dysregulated when you don't have these neurons. So one of the things we did is we tried to see, can we rescue these mice by giving them type 1 interferon? And that's one way by which we can kind of restore the homeostasis that you might see in a vehicle-treated mice. But then also we want to know what's the overall role of these immune cells in nociceptor-ablated mice. So one kind of broad way of targeting them is to deplete them. So first, just to confirm what we found by single-cell RNA-seq, if we did flow cytometry of the bronchial lower lavage fluid from these 2 RTX vehicle mice, you see again this huge influx of neutrophils and monocytes at day 7 that is not there in the control mice. So to target them, what we did is we gave the mice this antibody GR1, which will kill off both monocytes and neutrophils. You can see here the neutrophil influx is completely gone because we've ablated them. So what happens in terms of survival? So here you could see over 3 cohorts of mice so the vehicle-treated mice is here in blue with isotype control antibody, and then the RTX do a lot worse. But then if you kill off the neutrophils and monocytes, you can restore their survival, and also there's also somewhat benefit in the vehicle-treated mice as well. So just to kind of summarize, we think that if you don't have these protective nociceptors, then you get this increased viral spread and this huge influx of immune cells. So really the neurons are there to control immunopathology. But what I'm telling you here is it's context dependent. So the nervous system, or the peripheral nervous system, is a rheostat, right? So in bacterial lung infections, this same neutrophil control is bad because those neutrophils are needed to kill off the bacteria. So if you get rid of the neurons, there's a worse outcome. Sorry, if you get rid of neurons, you get a better outcome of infection. But with viral infection, these neurons suppress viral-induced inflammation, cytokine production. And I think there's still a lot of questions of how the mechanisms work. So is this through a body-brain axis, or is this through local neuroimmune control? But I think understanding how the nervous system regulates the immune system, we might need lead to better treatments for both pain and also infection and inflammatory diseases. So all this work was really carried out by amazing people in my lab. So Nicole Almanzer, who really led the flu work, as well as former lab members Kim, Pankaj, Swalpa, and Jingya. And some of the gut work was carried out by, again, close collaboration with Christophe Benoit, Diane Mathis, and others. So thank you for your time. Happy to take questions.
We have time for 2 questions. So I will get started with one.
Okay.
So it seems that this TRPV1 plus neurons protects against cytokine storm by suppressing inflammation, right? And as you know, many of these patients who survive cytokine storms they actually end up dying of immunosuppression. So I wonder, what's the kinetics of TRPV1 neuron activation, and how long do they stay hyperactivated?
Yeah, it's a good question. So, we've looked at actually a marker of their injury, not so much activation, called ATF3,
Okay.
and that goes up pretty quickly after viral infection, maybe within a day. The whole course of infection, though, takes about 10 days or so. They start dying around day 7. So it could be that even though they're responding early, the effects that there are to control inflammation start kicking in, and you need that balance, right? Because with resolution of inflammation, it's important that you have that happening, even from the beginning. So it's a complex. You're right. The kinetics is very complex.
Yeah.
The bacterial infection, we saw actually the control happen right away, and the bacterial infection, the mice die within 24 hours. So it could also be somewhat related to that, in addition to just.
I just wonder if you have a mouse model of septic shock, for example, and if it enters the second stage of the immunosuppression, perhaps these neurons are still activated, and that could be another way to target that.
Yeah, that's a good question. We haven't seen whether they suppress sepsis. Kevin Tracy is another person in this field. He's found that the vagus nerve, if you stimulate that, can suppress sepsis and TNF production in a very acute sepsis models.
Right.
I was going to ask a very general question. It was a great, amazing work. I was going to ask, what do you think the mechanism is through which the neurons are signaling to the immune? I'm just having a hard, like it's incredible. I'm just trying to understand, like, how does the activity of a neuron talk to the particular immune cell? Because which are floating around, right? The neurons are in a fixed position, and you have floating, circulating immune cells. So I'm wondering.
Yeah, so that's a great question, and that's still unanswered for the last part. For the first part, we think the direct communication with CGRP, because they are juxtaposed in the gut. But the second part, there are mechanisms, like to the point about systemic, where innervation of the spleen, for example, where a lot of these immune cells rush in from the spleen or the bone marrow into the lungs. So there's innervation of those organs, and I wonder if our neurons are actually triggering a circuit that goes to those organs, and then they suppress the exit of immune cells, or suppress. So these are, or it could be through the HPA axis, which is another classic way by which brain can affect the body.
Thank you for your great talk. My own study focusing on how microbiome metabolite influence the neuron circuits. So I was wondering whether the microbiome metabolite can influence the TRPV1 activity.
Yes, that's a very good question, for sure. So we've only so far looked at things like toxins, which can act on the neurons from the microbes, but definitely they have short chain fatty acid receptors, and they have AHR receptors. So I haven't seen any papers that have addressed that, but it would be interesting, right? Because that could affect pain, or block pain, one way or the other. So it'd be great to collaborate if you want.
Thank you.
Great. I have a question regarding you observe increased level of CGRP release after chemogenetic activation. Do you think the activation of neuron induce influx is sufficient to induce neuropeptide release?
Yes. Yeah.
Specifically, the release of CGRP impact on other groups.
Oh, I see. So definitely the CGRP and other neuropeptides are in dense core vesicles, and they're released by classic snare-dependent. So calcium influx in the nerve terminals will induce their release. And you may not even need an action potential. You can have just low levels of calcium influx is sufficient, because we found if we get rid of the neurons in the gut, there's actually lower levels of CGRP at homeostasis without any pain going on. So I think they're being tickled and calcium influx is causing that. So they store other things like glutamate and substance P and many other mediators. So and also my colleague Uli van Andrian has found they produce chemokines. So how all of these things are, what is being produced in each of these tissues by nerves is something very interesting to ask, like especially at different conditions like infection or. I think so. I think we can tune that, right? So if we give more CNO or, but there's a recent paper, like, so using optogenetics versus chemogenetics, there might be differences in the amount of calcium and other things. So I don't know for sure. I'm not Ed. So we can ask like how much, but it's a tuning thing, right? If you can increase the threshold, you might get more release. Yeah. Yeah.
All right, thank you.
Thank you so much.
All right, so we are gonna have a coffee break and then we'll come back in 20 minutes-ish. Doesn't matter. 我们回来。
没事，往下抠一下，因为
以后需要很多领事，对，很多对。高丽。
Oh, yeah. Join, right? No.
The email.
Yeah.
我介绍完走下去到那边去应该没问题，我在这看不到。
行。
对，我怕等会要问问题。
So, yeah, I just stay here, I guess. So.
Yeah.
All right. All right, everyone. Welcome back. Welcome back. So our next speaker is Dr. Jonathan Lipton, assistant professor at Harvard Medical School and Boston Children's Hospital. So Dr. Lipton is a pediatric neurologist and sleep specialist. He works on how circadian rhythms regulate brain health. His research has revealed novel non-canonical roles of the so-called clock proteins in synaptic function, cellular stress response, and neurodevelopmental disorders. Today, I thought he was going to discuss how circadian timing may hold potential for therapeutic intervention, but he just told me that he's going to talk about basic biology, which is actually more exciting for many of us. So please welcome Dr. Lipton.
Thank you. Thank you so much. And thank you for the opportunity to be part of this really cool symposium. I think it's really a amazing testament to this community. It's just the incredible breadth and quality of the talks today. I mean, I've learned a lot. It's just been really quite amazing. And I think it's just, again, I know a lot of the other speakers have mentioned this, but I think this is just a chance to remind ourselves that we're here to, you know, shine light and find discoveries and help the patients in this around us eventually. And I think we just have to remind ourselves of those goals because they're important, and nothing should deter us from that. All right, I'll get off the that part now, but I just wanted to say that. Let me just make sure this is working. There we go. Oh, this is just a quick disclosure. Nothing really that important. So yeah, you know, so I always start with this slide. This is from a movie by a Swedish director named Ingmar Bergman. And this is a really cool movie called Wild Strawberries. And basically this elderly gentleman goes to sleep and he then wakes up in his hometown and he looks at the clock at the hometown and it has no hands. And this sets off the premise of the movie, which is basically a crisis. He goes through a whole crisis about his life and, you know, this existential confusion about what his life is about and where it all went. And I think I like this because it just helps remind us what time is about and what timekeeping is about. And what time is really about is prediction. So if you go to any city, really, any town, there'll be a clock of some kind in the center of that town because we all rely on this fundamental process to help us predict what's going to happen in our day and in our life. So for example, they have this beautiful clock in Prague, Big Ben. This is actually a Chinese sundial. And then the famous flower clock, which was one of the original demonstrations of an internal timekeeping systems and living systems. So just to remind us, one of the most fundamental aspects of our life on this planet, besides the fact that it has gravity and keeps us actually attached to the ground, is that it rotates. And as a result of the rotation, we experience a geophysical oscillation of the light-dark cycle, which really imposes this predictable rhythm to the sources of energy, which really ultimately come from photosynthesis in plants. And so as a result, pretty much all organisms across all 5 kingdoms have elaborated a clock or timekeeping mechanisms that help those organisms synchronize with this reality that we live on, which is a oscillation of our globe. And so all kingdoms have some form of a circadian timekeeping mechanism, and those mechanisms are really similar to the foundation of any oscillating system. You have a positive limb, so something driving something, and then you have a negative limb, so like a wave, you know, something making something go up and then something going down, and then this is iterative and repetitive and really fundamentally autonomous. So pretty much clocks of any kind and clocks in all systems are built on this oscillating clocks are built on this negative feedback loop, whether it's in a sporulating mold or a human being. Okay, so the human circadian system, the circadian timekeeping system, really can be understood. I'm going to divide it. There are many ways of showing this, but I'm going to show it as sort of like 3 steps. And of course, I'm going to use music because I'm a musician, so I'm going to use music as a. So really, you have the hierarchical organization. You have the hypothalamus, which has a tiny little group of neurons called the suprachiasmatic nucleus, which form a central autonomous oscillating system that has a 24-hour period, roughly 24-hour periodicity, depending on species and genetics. And that acts as like a conductor, you could say as like a conductor. You then have, the conductor has to conduct something. Sorry, I wonder if anybody knows what piece that is. That's actually Beethoven's 5th Symphony, the beginning. And as a conductor, I can tell you the thing that's so hard about conducting that piece is that it starts with a rest. But anyway, so there's the thing that actually the conductor is conducting, which is this internal timekeeping system, this clock mechanism, which we'll discuss in a moment. And then, of course, there's this output, which here I have as the orchestra. So the timekeeping actually has to be manifest by the playing of the music or the playing of the time-related signatures. And we're going to discuss that in detail. So just to review this fundamental mechanism, sorry, the animation got a little messy, so ignore this for a moment. So the timekeeping system in mammals is rooted in this negative feedback loop, which in 2017 was awarded the Nobel Prize to Mike Rosbach, Mike Young, and Jeff Hall, who cloned the Drosophila period gene, and that led to the expansion of the entire system. And basically what we have here is we have a transcriptional translation feedback loop, or TTFL, in the field. And that positive part of that in green is the BMAL1 and clock transcription factors that form a partnership and drive many, many thousands of genes, including their own inhibitors, the period and cryptochromes, which go out into the cytoplasm, go through all sorts of machinations and regulation, and then come back in and turn off the things that turned them on in the first place. And so this is a positive, negative clock that remarkably takes about 24 hours to complete itself. And depending on the cell type or condition, this can regulate anywhere from 10 to 30 or sometimes even more of all genes. So sorry, yeah, the animation got a little screwy. So this is just to show you the one component of this system that is non-redundant is BMAL1. And so on the top, we have here wheel running data from mice. So mice are nocturnal. So in the dark, they'll start running on wheels. As you put them in all dark conditions, they'll continue to maintain a regular period, although that's a little bit shorter than 24 hours, about 23 and a half hours. This is some light pulse added in here. In a BMAL knockout animal, you just have a disorganized pattern without any circadian rhythmicity at all. And that's true in cells too. So if you, in black here, if you remove BMAL from cells, you lose this internal oscillation of the cellular rhythm. And this is the point I wanted to make, is that the clock is a multi-scaled system that exists at the level of molecules, cells, tissues, organs, and complex behavior. Like, you know, for example, we were all just standing in line for coffee. Like if one were to, you know, model this, it would have a circadian pattern, you know. And I was making a joke before that I always get to speak, I always seem to speak in the mid-afternoon, which is the dip in our level of arousal. It's called the circadian dip. So I hope you all had some coffee. Anyway, what I wanted to bring out is this idea of why this matters. Why do we have this prediction system? And one of the reasons is that it helps, it's one of the primary homeostatic systems in the body. So one of the things it does is maintain equilibrium. So this is a very, very rough model. If we imagine that health occurs, if the green lines represent sort of the normal range of a healthy oscillation of some variable, let's say blood pressure or heart rate or your gut flora, something like that. We can imagine it oscillating day by day, and we can imagine then some perturbation to that. Like, let's say you, I don't know, you eat something different, or you're a little bit jet lagged, or you have some medication or something like that. You can imagine that it would change that, but still stay within physiologically acceptable levels, that it doesn't cause stress. And we would say that that's like maintenance, or like, the system is maintaining health or maintaining equanimity. Now, we can imagine where timing could be really important would be like, for example, when the oscillator itself or the timing of the oscillator is shifted significantly, suddenly there are particular times of day where that threshold is now dangerous. You're now outside of the resilience period, and you're now in a pathological period. And while it's hard, there are not a lot of direct examples of this. There are some. And I think the important thing I wanted to raise here, I don't have time to get into all of it, is that the ubiquity of this system in all cells pretty much of the body, and its control of many, many systems in the body is also part of the reason why it seems to show up in all major disease processes. So when the clock is disrupted, here I'm just showing a neurological disruption. We have pretty much all the major classes of neurological disease have been linked to circadian disruption, or can cause a disruption of circadian rhythms. And that includes intellectual disability, stroke, neurodegenerative disease, epilepsy, and even neuropsychiatric disease. This is, of course, not exclusive to neurological disease. The clock has also been linked to cancer, inflammatory disorders, and certainly metabolic disorders, just to name a few. And then, and of course also cardiovascular disorders. So this is a key homeostatic system. I'm a pediatric neurologist, and my intro into this entire field came from my interest and also my exposure to children with neurodevelopmental disorders. I was in my residency here across the street, I was doing in clinic, and I would have patients with autism who couldn't speak, and all complex neurodevelopmental problems and genetic disorders. But the only thing that the parents wanted to talk to me about was the fact that their kids weren't sleeping. And so as a result, they weren't sleeping. And this is really the main thing that was like tearing these families apart. It was really ruining the quality of life. And to be honest, I had nothing to tell them. I had nothing for them. So I decided to start looking into this, and I ended up meeting became my postdoc mentor, Mustafa Sahin, who was studying a neurodevelopmental disorder called tuberous sclerosis complex. So, so sleep dysfunction is incredibly common. We say it's a bedfellow of neurodevelopmental disorders. And this is just a very small list. The list is much, much longer now. We have a lot more insight into this. But at the time when I started, this was more than 10 years ago, or 15 years ago, we really didn't know anything about what TSC might be doing. And Mustafa said to me, he said, well, these kids have horrible sleep. What can we do about that? I said, I have no idea. Let's look into it. So we started thinking about ways to approach this. And I'm going to summarize, many years of work quickly. But what I'm going to tell you is that what we discovered is that the cause of TSC is actually directly interfacing with the circadian clock. So what is TSC? TSC is part of a fundamental pathway, which many of you, all of you, I'm sure have heard of, called the mTOR pathway. TSC is an inhibitory gap complex that basically is an obligate inhibitor of mTOR. It integrates tons of signals upstream. And what I discovered is that I discovered that in TSC mutant cells, the output of the circadian clock was markedly abnormal. And I subsequently went on to show that the reason for that is that when TSC is lost, we have a disinhibition of mTOR. mTOR levels are very high. And what I discovered was that the BMAL protein, which is this key for the circadian rhythm, is markedly dysregulated. And it's dysregulated on 3 levels. Number one, I found that an mTOR effector kinase, either S6K1 or AKT, can phosphorylate BMAL at one site, which is actually very important, which we'll discuss. The second thing is that mTOR will dysregulate the translation of BMAL, so the amount of the protein gets made. And then mTOR also regulates an axis of the proteostasis of BMAL. So there are these 3 layers. We have the turnover of the protein through ubiquitination, translation of the protein, and phosphorylation of the protein. All are dysregulated. And as a result, we have phenotypes in mice. So these are global heterozygous mouse. And what you can see here is that these mice, in response to a jet lag, they phase shift very quickly. And because we showed that the BMAL levels were high in these mice, we had this crazy idea to knock it down a bit. So we made a double heterozygote where we have the amount, and we actually were able to rescue both a free running period, so the endogenous circadian rhythmicity, and this jet lag phenotype, suggesting that the clock is actually indeed a downstream pathway. We've subsequently went on to look at a more severe model. This is a knockout of the TSC gene in neurons, and this is known to be lethal. When we knock out one copy, we knock out 2 copies of BMAL in conjunction, we're able to almost double the lifespan. We don't rescue completely, which we wouldn't have expected, but we are actually able to show that we can get some degree of rescue, again, supporting the idea that BMAL is a downstream component of this mTOR pathway with relevance to TSC. So what this kind of like led us to think about is that, we seem to have this core clock machinery that's kind of in this, I mean, this is a little artificial, but it's sort of like we take together these data points. We have 3 different nodes. We have the TSC mTOR pathway. Sorry, I'm going to go back for one moment. We have this ubiquitin ligase called UB3A, which is responsible for degrading BMAL. And that protein is actually a cause of a disorder called Angelman syndrome. We also have a deubiquitinase, which can deubiquitinate BMAL, called USP9X. This is cause of another X-linked intellectual disability and a seizure disorder. And so we can start to think that, this clock machinery is somehow a node that's linking together, maybe downstream, these different neurodevelopmental disorders that have a variety of unique but also overlapping features. One of the things that is going to be the rest of my talk is really the fact that all of these disorders cause synaptopathy. These are disorders of synaptic function. And because I had discovered this phosphorylation event in the BMAL protein and a role for it in the cytoplasm, we started thinking about, well, could this protein actually have a role in synapses? And to characterize that in a broader sense, what we really want to think about is how we go. So when the fundamental role of the brain is to carry out computations, obviously, to allow us to do everything that we do. And in thinking about this from a circadian perspective, we have a little bit of an issue because we have the TTFL operating between the nucleus and the cytoplasm, regulating a number of genes over very slow time scales. And then we have synapses dotted throughout the processes of the neuron, this like the ultimate cytoplasm, right? This incredibly elaborated, reified cytoplasm, where we have synapses throughout communicating sometimes tens or hundreds of thousands of cells. And so the question we wanted to ask is, could there be a role? How is circadian information integrated across this spatial temporal divide between a transcriptional system and a spatially separated system, in addition, the temporal separation between the very rapid changes that have to be integrated by synaptic function with this slow oscillation of circadian rhythms. So one of the things we found when I first started my lab about 7 or 8 years ago, and this came somewhat by accident, but we discovered that the BMAL protein, and I don't have time to show you all the data, but the BMAL protein basically is expressed in synapses. So it is a transcription factor, but we find that it's actually localized to synapses. These are synapses of the hippocampus, but we also find it in cortical synapses as well. So not only that, but it's also rhythmic in synapses. So if we take the animal, we take these immuno-EM images from the morning or the evening, we can see a rhythm to the amount of the BMAL in the synapses. And when we asked, so what is it doing there? We did pull downs for the endogenous protein and then like sent it for mass spec. And the number one protein that we got out is a very abundant protein in the brain called calcium calmodulin dependent kinase 1. So CAMK2 alpha. So CAMK2 alpha is the brain's most abundant kinase. And on some level, we were like, oh, that's kind of interesting. You know, CAMK is involved in circadian timing and sleep and plasticity and long-term potentiation. So like this could be exciting, but it's like abundant. So we're like, oh, I don't know, maybe. Well, let's see. But let me just tell you, it turns out to be absolutely crucial. So let me just tell you a moment about how CAMK2 works. CAMK2 is an incredibly interesting and super complicated enzyme. It basically is made up of each one of these is a subunit. There's a hub domain, there's a regulatory domain, and a kinase domain. What happens is when calcium comes into the cell, it binds to calmodulin. Calmodulin comes onto the regulatory domain and opens the protein. This protein exposes the kinase domain, which can now phosphorylate its neighbor. It phosphorylates it at a single residue called T286, which keeps this protein open. And then because this protein forms a multimer, actually a hexamer, a two-part hexamer that forms a 212mer, it's a dodecamer actually, it can take this calcium transient, which goes usually in the order of 200 microseconds, and it can translate it into a very, very long biochemical event. So it basically it translates time from a microsecond scale to minutes or even longer scale. It's literally like a biochemical computer for calcium. CAMK has many substrates in the brain, including very important disease and synapse-associated molecules like synapsin, SYNGAP, and NMDA receptors. It's involved in crucial functions that I've already mentioned. And one of the things I want to mention, which is crucial, is that it's involved in both postsynaptic and presynaptic function. Okay, so without getting into the lane-by-lane information of these immunoblots, I'll just tell you what they show. These are immunoprecipitations from synaptosomes from mouse forebrain in both directions, usually either BMAL as a bait or CAMK as a bait, and basically shows that the proteins can interact. We've done a whole host of other cellular assays to show that the proteins interact, and they interact directly, even in vitro. And I'll just show you this this is it was a bit of a crazy part of the story. So when we found that BMAL associates with CAMK, we were like, oh, well, maybe, you know, a generic thing would be that it's a substrate of the kinase. It just gets phosphorylated by the kinase. We actually cannot detect any transfer of phosphate to BMAL from CAMK whatsoever. On the other hand, what we do see is that when we add BMAL to in vitro assays or in vivo, as it turns out, we actually can, we see potentiation of the T286 site. So potentiation of the key event that keeps the kinase open. So BMAL regulates the kinase, okay? I will get back to that in a moment. So we've now done a lot of mapping to try and understand this. Without getting into all the details again, I'll just say on the CAMK side, we know that CAMK needs to be in an open state for the BMAL to come on with it. But what's really fascinating though is that we can then we can really be vigorously wash this and it still stays on even after calcium and calmodulin come off. So once BMAL comes on, it keeps this open state and we think it helps potentiate that state. We've mapped that to the kinase domain itself, but what's interesting is that it binds outside of the substrate binding region, consistent with the fact that it's not the substrate, not a substrate. On the other hand, BMAL, so BMAL has, it's a BHLH domain, pass domain protein, actually in the same family as HIF2 alpha, which we heard about this morning from the father of the field, which is kind of cool for me. It's a BHLH pass domain protein that has both two disordered regions, one on each side. We found basically that neither of these have anything to do with binding. And interestingly, this is this phosphorylation event that I found, which is going to play a very important role in the rest of the talk. So we'll hold on to that. But it turns out it binds through here. We ran simulations with AlphaFold, like everybody, and I think these are just simulations, so we take them with a major grain of salt. But it does show us something interesting. If we run the pass domains on AlphaFold versus the BHLH domain has a much higher predictive accuracy. And what's interesting is that's shown here in orange. It binds to this this fold of the P286 site. So very consistent with the fact that we only see open forms of the kinase binding the protein. And the other thing is if we do three proteins together, so here we did SYN1, synapsin1, which is a classical target of CAMK, the SYN1 will fit very beautifully into the substrate binding pocket right here, and BMAL is next to it. So overall, we think this has face validity, and we're now trying to validate this structural part of it. Okay, so what I wanted to show, get to now is what regulates this timing of BMAL in the synapse. And I had previously discovered, again, this got screwy here. I previously discovered that the mTOR pathway regulates the phosphorylation of BMAL at this one site. And what I found is that that actually regulates protein synthesis downstream of the mTOR pathway. So it can actually regulate rhythms of protein synthesis. So when I started my lab, one of the first things I wanted to do is knock out this site to see what happens in vivo. So we did that. And actually, when we manipulated this site in cells, we never saw any change in the transcriptional output of the clock, only in protein synthesis. So when we ran these mice that now carry a single point mutation in BMAL1, they have actually completely normal wheel running behavior. And that tells us that the SCN, the central clock, is working. And it tells us that the TTFL mechanism, the transcriptional mechanism, is working, because both of those are required for organization of behavior. But what's pretty, so just in contrast, just to remind you, to the BMAL knockout. So the BMAL knockout has no rhythms. These guys with the point mutation have intact rhythms. But what gets interesting is if we look in the synapse and we see, look at the rhythm of BMAL in the synapse. So here, BMAL high, low, actually it's there just very low. But in this mutant, the rhythm is completely gone. And so the rhythm is just high, it's just there all the time. And in fact, we see this for across, not just for BMAL, but for CAMK, CAMK's associated proteins, and for outputs like long-term potentiation. So here, just don't worry about this so much, I'll show you here. So this is the baseline change in long-term potentiation. So we recapitulated work from other groups showing that there is actually a rhythm to long-term potentiation. It's higher in the morning compared to the evening. But in our mutant, there's a slight effect, but it's completely not significant. So again, even though the global behavior of the mouse is normal, this cuckoo, the global behavior is normal, the difference between the morning and evening at the level of the synapse and synaptic function is lost. So we put together a model where the BMAL under the normal conditions is phosphorylated. That phosphorylation is regulating the amount of BMAL in the synapse, which in turn is regulating the amount of this CAMK. And we think what it's doing is it's setting a dynamic range of plasticity between the morning and the evening. When BMAL is not phosphorylated, it basically can't exit the synapse. It sits there potentiating CAMK, keeping it locked in a particular state. And as a result, we end up with a loss of dynamic range. It's almost like, to have a dynamic range of plasticity, you need to move from either the floor to the ceiling or the ceiling to the floor. You can't be stuck on either end. If you're already at the top, you can't go anywhere. And if you're at the bottom, you can't go anywhere. So what we think is happening here is that is the BMAL protein, this clock transcription factor, is repurposed to the synapse to tune a fundamental organizer of synaptic function. And this work was all led by Ilaria Baroni, a super talented postdoc who's now in Italy, her own group, and an MD PhD student who's carried on the structural work. So how much time do I have?
5 minutes.
Okay, I'll go fast. So so that's all been published. We now are working on the presynaptic side. We asked, well, we know that CAMK is crucial for presynaptic function. We see BMAL on both pre and postsynaptic sides. And when we look at the BMAL, we actually see it very close to synaptic vesicles. So synaptic vesicles, just to remind the non-neuroscientists, these are the membrane-bound bodies that hold a neurotransmitter and are actually basically the essential component for neural computation. So they actually are highly regulated. It's actually besides protein synthesis, the most energetically demanding process in the entire brain. And what we find, okay, so in our wild-type mice, we see these are mini postsynaptic potentials. So we see postsynaptic currents. We see a normal amplitude on the postsynaptic side, but an impaired frequency. These are single cell recordings done by my colleague Hisashi Umemori. And we see in our mutant a very impaired frequency. So a putative reduction in release. And that seems to pair out because when we do something called pair pulse facilitation, which is a way of challenging the presynaptic side to ask, as it's basically a proxy for the neurotransmitter release frequency, we actually see it's higher, which actually, for those of you who are not used to interpreting the physiology, means that the vesicles are not being released. So that suggests an impairment in the vesicle compartment. So then when we went back, we did EM on these animals, and we did all of this completely blinded. And what we found, we actually counted by hand 70,000 vesicles in the hippocampus. And what we found is that the vesicles in the point mutation mice, remember these mice lack a rhythm of BMAL in the synapse. There's just chock full of vesicles, chock full. It's actually much higher. And actually, even when we take the cells from these mice and we culture them, we can actually see higher levels of the vesicle pool protein synapsin, but no change in the actual active zone. So no change in this number of synapses, but a huge increase in the vesicle pool. And then what we did is we did this, of course, in time. So we did the same experiment in circadian time. And what we found is that there's a rhythm, a diurnal variation between the morning compared to the evening, highly significant. And again, in our mice, it's just completely gone. And this is just showing that the vesicles are all crowded and stuck together. So we think that the BMAL protein is organizing a synaptic vesicle dynamic of some kind. And that bears out with biochemistry as well. CAMK, one of its primary targets in the presynaptic site is the synapsin protein, and this is a CAMK specific site. That site has a rhythm, whereas in our point mutation, that rhythm again is gone. So the last thing I'll say is that I showed you that these mice do not have any circadian phenotype. They do have a learning phenotype, so they have impaired fear memory, which is a hippocampal dependent behavior. And then we've subsequently found, I'm just going to skip to this, that they do also have an interesting sleep phenotype. So here we've done sleep recordings, and what happens is what you can see is that the mice, the purple is the mutant, they peter along fine, they wake up at the right time because their clock is working. The timing for waking up is fine. But then after they wake up, they fall back asleep. So this is non-REM sleep. They fall back asleep more during the first 6 hours. Now it's a relatively weak effect. It's not super strong, but it's very significant and seems to be reproducible. And what's interesting about this is that this is, to our knowledge, one of the first examples linking together the timing of wakefulness to the circadian system itself. So something that actually regulates the timing of maintaining a sleep-wake state, dependent on the regulation of the clock protein. Not just the organization of sleep-wake states, but maintaining the actual vigilant state. Okay, so what I've told you is that the BMAL protein is found in synapses. It has a rhythm dependent on its phosphorylation, which seems to organize synaptic plasticity, memory, and sleep, among probably many other things. And we believe that it has roles on both postsynaptic and presynaptic side. The postsynaptic part is published. The presynaptic side, we're currently writing up the manuscript, and hopefully that'll go okay. And then I'm just going to summarize here. The circadian system may be a node for neurodevelopmental proteopathies and synaptopathies, and this is obviously become a much deeper story and something we're really interested now in pursuing in a variety of different models. The protein rhythmically localizes, as I've suggested. BMAL is a biochemical organizer of CAMK, and we think it might be a novel organizer of CAMK because there's no other example of a CAMK modulating protein that we're aware of that binds outside of the substrate binding pocket. So we think this actually might be novel. Rhythms of BMAL phosphorylation permit a rhythmic efficiency of synaptic plasticity independent of the core oscillator, independent of the TTFL. And then our hope is to now apply this to a variety of disease systems. So one of the things we have a collaboration with a colleague at Mass General working on a traumatic brain injury model that invokes this BMAL phosphorylation mechanism and disrupts actually the timing of the synaptic rhythm. So we're really excited about potentially applying this down the road. Most important slide. I hope I spelled this correctly. But thanks to all of you. Thank you to my lab. Ilaria Barone really spearheaded all the work most of the work I showed you, and helped by many other folks, including super talented MD PhD student Nicole, and a variety of research techs, including Jack, Engie, and Yana, and former members of the lab who were instrumental funding, and my collaborators, especially Peter Tsai, UT Southwestern, who helped us with all our behavioral assays, Hisashi, for all the physiology, Michael Whalen with TBI, Meg Stratton, who's a CAMK2 goddess, who's been really helping us with the structure function analysis. And yeah, that's all. Thank you.
All right.
Hi, Jonas. Wonderful talk. So my question probably going to be a little bit naive because I'm not a neurobiology person. I'm a metabolism person. I'm very impressed to see the mTOR really play a role in this circadian behavior regulation. Because in the field of metabolism, mTOR is a master sensor for nutrients and energy status. But I mean, you didn't really tell about what is upstream signaling can really matter this like a circadian regulation. Okay, here's my question. So I feel I always got like sleepy after a meal, especially lunch. I think it's happened to a lot of people. So I'm wondering what is the signaling really matter this process? So now after listening to your talk, I'm thinking maybe after a meal, our insulin level will go up, right? Because to lower the glucose level. And then I would assume the mTOR signaling is activate. So do you think it's actually the insulin mTOR signaling is really mad at this process? Makes me sleepy after the, especially lunch, yeah.
Yeah, that's a you want to know everything. It's good. That's really good. It's really good to like have a huge model for the whole system. Yeah. So, okay. So the mTOR pathway is crucial for circadian rhythms. It's a crucial output. It actually has the pathway has circadian rhythmicity that's partially independent of the core oscillator, so probably related to other factors upstream, such as signaling through growth factors, insulin, IGF-1, absolutely essential synchronizers of especially peripheral clocks in the liver and things like that. So in terms of the sleepiness after eating, there were probably a whole host of reasons for that, which I won't get into. But one thing just to mention, so to help you understand, is that the body is synchronized. The entire body is synchronized, right? And our behavior is organized into sleep-wake cycles by the day. But the tissues in the body are not all in the same phase. So they're synchronized, the periodicity is synchronized, but the relationship of the signaling is dependent on the tissue. So the liver and the heart are a little off and they're not necessarily in synchrony with the brain. And in the brain, you can get into all sorts of things about like, because the brain is not one organ, it's like 1,000 organs jammed into the skull, right? So it can get very complex on that level. So that probably has, I hope that helps you think about it. Yeah.
All right, thank you.
I'm supposed to hold it. Okay, good.
All right. After we figure out full comma, let's move on to our final speaker. It's a privilege to close our section with a keynote from Dr. Li-Huei Tsai, Director of Picard Institute for Learning and Memory and Professor of Neuroscience at MIT. So Dr. Tsai's research has been instrumental in uncovering molecular mechanisms behind neurodegeneration and memory loss, ranging from her early work on CDK5 mediated neurotoxicity to the recent invention of Genus, a non-invasive approach that uses 40 Hz sensory stimulation to reduce amyloid plaques, tau tangles, enhance brain clearance, and does many other things in the mouse models of AD, and I believe is currently in the clinical trials. So we'll hear about this today. Dr. Tsai is a member of Neuro Academy of Medicine and continues to push the boundary of translational neuroscience. Please welcome Dr. Tsai.
Hello, everyone. Thank you for staying until the very end of the day. It is certainly an honor for me to speak to you about our ongoing effort on trying to understand how promoting brain synchronous neuroactivity can provide neuroprotection. So before my talk, I just want to disclose that I'm a scientific co-founder of Cognito Therapeutics, a biotech company currently carrying out phase 3 clinical trials on the technology I'm going to tell you about. So, so we know that the human brain contains billions of neurons, roughly 87 billion of neurons. And these neurons obviously connect with each other to form this very complex and intricate network and circuits through the formation of synapses. So the information in the brain is processed via neuronal firing. However, synchronous firing of neuronal ensembles is believed to underlie behavioral states. So the synchronous firing activities can be detected as brain rhythms or brain waves, as popular science would like to call it, by electroencephalogram or such as EEG or other electrophysiological approaches. So in the mammalian brains, there are more than 10 different classes of brain rhythms that can be detected. And these different classes of rhythms, they differ by orders of magnitude in frequencies. So, so these frequencies go from infra-slow to the ultra-fast. And as I said, these different classes of frequency strongly associate with different behavioral states, such that the slower wave, such as the delta wave, is more active during sleep. The theta wave is active during memory and navigation. But I really want to talk to you today about the gamma band synchrony. So gamma oscillations ranging between 30 to 80 hertz is known to be the highest during higher order cognitive function, such as attention or decision making, formation of learning and memory, and so on. And at the neural network level, gamma band synchrony is implicated in gain modulation, network inhibition, phase coding, in essence, basically the computation of the neural network. So while we know a lot about when gamma is active and what's the associated network activity, very little is understood in terms of how these different classes of synchronous neural activity impacts the neural system at the molecular and cellular level. Okay, so we don't know how neurons and other cell types in the brain respond to gamma oscillation and whether they actually play an active functional role in mediating gamma effects. So today, I hope to provide some insight into these questions. And we started asking these questions by through working with Alzheimer's dementia, which my lab has been focusing on for the last couple of decades. So we know that Alzheimer's disease is pervasive and devastating. And it not just affects cognitive function, but it also creates huge burden for caregivers and family members. So the disease has characteristic pathological features, including extracellular amyloid plaques and intracellular tau tango pathology. But it is also known to cause brain volume loss, synaptic loss, neural inflammation, and so on. But Alzheimer's disease also has a huge impact on the neural network activity. And it has been shown that in the early stage of the disease, when amyloid accumulation starts to appear, there is neuronal network hyperactivity, so increased firing of these neurons. But later on, after tau pathology emerges, then the network instead adopts hypoactivity, so reduce neuronal network activity. And in particular, gamma band oscillation has been shown to be compromised in Alzheimer's disease. And this can be nicely summarized by a review article by Jorge Palop and Lara Milky at UCSF, where they show using the schematic that when there is neuronal hyperactivity, there is reduced gamma band oscillation power. Whereas when there's increased gamma band oscillation, there is reduced network hyperactivity. And this has been shown not just in mouse models of Alzheimer's disease, but also in human patients. And in our own hand, about a decade ago, through collaboration with Boyden's laboratory, we wanted to see whether the gamma band compromise is an early event. So we work with pre-symptomatic amyloid mouse model 5-FAD animals. And we actually perform local field potential recordings in the hippocampus of awake behaving animals. And there we were able to show that gamma band oscillation centered around 40 hertz is already reduced in this early stage 3 months old 5-FAD mice compared to the wild type littermate. So, so back then, a graduate student, Hunter Alcorino, asked a very, very simple question. So they wanted to know whether boosting gamma activity will have any measurable consequence. So they team up with Annabelle Singer, at the time a postdoc in the Boyden lab, and we use optogenetics using channel rhodopsin 2, as you heard from Ed, introduce the channel into the hippocampus of the PB creep class 5-FAD. So, so this way we can use laser pulses to activate these pulvalbumin neurons at 40 hertz. And we found that this is sufficient to drive increased 40 hertz oscillatory power in the hippocampus. But then next, they made some very surprising result, which is after 1 hour of activation, they found that amyloid beta peptides 1 to 40 and 1 to 42 show profound reduction, okay, just after 1 hour by about 50% following 40 hertz stimulation. And this was not observed with 8 hertz or random frequency stimulation. So with these results, I just want to cut the story very, very short. So given that optogenetics is rather invasive, we eventually figure out to use an extremely non-invasive approach to manipulate the firing patterns of neurons in the brain, basically using sensory stimulation. So we can just, you know, in a mouse in the home cage, presented the mouse with LED light, flash at desired frequency, and we found that neurons in the brain can follow this sensory patterned sensory stimulation to produce the same frequency of oscillatory activity. And later on, we also realized that we can stimulate other sensory modalities, such as using sound or vibrotactile stimulation to achieve the same effect. So, so we turn this approach gamma entrainment using sensory stimuli, or genus, when we present the simulation at 40 hertz gamma frequency. So, so this is just to quickly show you that so this approach is very powerful in activating neurons in not just in the visual cortex, but in many parts of the brain, including the hippocampus and the prefrontal cortex. And using local field potential recordings, we were also able to show increased 40 hertz gamma power in many different parts of the brain. So, so we quickly were able to show that with sensory stimulation, just 1 hour also, we were able to show reduced beta amyloid peptides in the brain, specific to the 40 hertz stimulation, not with 20 hertz or 80 hertz or random frequency stimulation. But very early on, we also realized that this stimulation not only drive neuronal activity, but it also can affect other cell types in the brain. Here, as an example, we use IBA1 to label microglia. And I think you can just see for yourself that the morphology of this microglia after 40 hertz stimulation is very different, okay? They look, you know, the cell body expanded and the processes look more complex, resembling a activated microglia state. So, so we have done a lot of follow-up work to show the sensory stimulation is able to reduce other Alzheimer's pathologies such as tau, hyperphosphorylation, and is neuroprotective, can preserve synaptic density, and also can improve cognitive behavior. And these are a few of the recent publications. We found that the sensory stimulation is really very beneficial in preserving white matter integrity. So here we use a chromosome paradigm to induce demyelination. So you can see this major white matter tract, the corpus callosum, show greatly reduced volume, but with chronic the audiovisual 40 hertz stimulation 1 hour per day for several weeks, we found that this is able to ameliorate chromosome-induced demyelination. We also use another method to induce white matter information using a chemotherapy agent, cisplatin, and this can induce so-called chemobrain pathology as well as affect axonal myelination integrity as shown here using transmission electron microscopy of the corpus callosum. You can see a lot of the axons are without myelin protection. But with the stimulation, you can see a restoration of myelination. And our most recent study showed that in a Down syndrome mouse model, which Down syndrome is caused by trisomy of chromosome 21 in humans, and these individuals show tremendous defects in brain development as well as embryonic and adult neurogenesis. And we found that, again, chronic audiovisual stimulation can lead to rather profound increase in adult neurogenesis in the dentate gyrus. And this is associated with improved cognitive behavior. So, so I think since our initial observation of using sensory stimulation to drive neurosynchrony and protective effect, we and many other laboratories now have shown very consistent neuroprotective effects induced by the stimulation, including reduced inflammation, neuroprotection, improve cognitive function, reduce pathology, and so on. And a lot of this neuroprotective effect is associated with robust glial response and response from the brain's vasculature. So what my laboratory has been focused on right now is really trying to figure out the mechanisms behind all of these beneficial effects of sensory stimulation. So we initially decided to start with, figure out what's the mechanism behind the rapid reduction of amyloid in the brain. With just 1 hour stimulation, we could observe such a reduction. So since we saw this very rapid microglial response, the microglia are known to be able to phagocytose amyloid, we wanted to know whether microglia play a role in this process. So a former postdoctoral fellow, China Daikan, decided to use a well-established drug, PLX 3397, which is a CSF1 receptor antagonist, and this drug can very potently deplete microglia in the brain. So, so he started out with very old 5-FAD mice, and then fed them with the PLX diet for 50 days. And here you can appreciate this diet led to about 60 to 70% depletion of ABA1 positive microglia in the brain. So, so the idea is that if these microglia are important for genus mediated amyloid reduction, then now when we deplete this microglia, genus should no longer reduce amyloid. And very surprisingly, this was not what we saw. So we compared amyloid plaque density before and after simulation, and with or without PLX diet. And we found that after PLX depletion, 40 Hertz simulation can still very profoundly reduce amyloid density, both in the cortex and in the hippocampus. So these results really indicate that microglia do not play a role in this process. So what else could be responsible? So now the well-established brain clearance system, in addition to microglia and the vasculature, is known to be the perivascular CSF influx and the lymphatic clearance of the metabolites, brain waste. So, so we know that CSF, which is produced by the choroid plexus, can move into the brain, and CSF travels in the perivascular space along the descending axon, descending arteries, sorry, into the capillary bed, where the CSF exchanges with interstitial fluid in a aquaporin 4 dependent manner. And this process leads to brain waste clearance. So this is a movie created by Michael Neidegar. I always feel that this movie very clearly demonstrates this process. So here CSF is labeled in green. It can travel down into the brain and then through the convective flow, clear the brain waste along the process. So we wanted to know whether this has anything to do with genus-induced amyloid clearance. So a former graduate student, Mitch Murdoch, set up a 2-photon real-time imaging system. So here he labeled the blood vessels in magenta and the CSF tracer in cyan, which he administered through the cisterna magna injection. So you can see over time, there is increased presence of CSF in the brain. So with this, he then compared the CSF intensity in the brain before and after 40 Hertz audiovisual stimulation. And what he found is that this stimulation profoundly increased the CSF intensity in the brain's parenchyma. And he was able to show that, again, this is a 40 Hertz specific phenomenon. It's not like any neuronal activity can increase CSF influx. And the mechanism mediating the perivascular CSF influx is known to depend on the dynamics of the artery pulsatility. So here is showing you an example. We are looking at the artery here, and you can see the artery really is very dynamic. Its diameter changes constantly. Okay, so this is known as arterial vasomotion. And Mitch was able to show that the sensory stimulation can robustly increase the arterial vasomotion, and 8 hertz and 80 hertz stimulation did not cause a similar increase in the vasomotion. And I want to bring up that this vasomotion is particularly prominent, the increase in the sub 0.1 hertz, a very slow vasomotion. And this kind of slow vasomotion has been particularly demonstrated to be important for CSF influx. So the question remains how the 40 hertz gamma stimulation leads to increased arterial vasomotion and CSF influx. So Mitch performed a single nucleus RNA sequencing transcriptomic analysis of the 5-FAD prefrontal cortical tissues after 60 minutes of stimulation. So, from this RNA sequencing result, we look at differential gene expression, and we found that the cell type that show the higher numbers of the DAX are the interneurons subtypes SST and VIP, along with astrocytes and the vasculature endothelial cells. Okay. And so when we particularly look at the gene ontology, that the biological processes that are particularly impacted by the stimulation, they turn out to be peptide synthesis and intracellular protein trafficking, membrane anchoring type of processes. So I just want to point out some of the top upregulated genes. And very interestingly, we found a number of neuropeptides, including VIP, SST, VGF, MPY, and so on. So to validate whether there's really increased neuropeptide signaling, at Boyden's lab developed a VIP sensor. So we were able to look at VIP signaling in vivo 1 hour after 40 hertz stimulation. And we saw this very significant increase in VIP signaling. So you know, this increased neuropeptide transcription seems to have a consequence. And another class of genes I want to point out are highly upregulated in astrocytes. And these genes are known to regulate aquaporin-4 trafficking and membrane localization. So as I mentioned, aquaporin-4 is a water channel expressed by the astrocyte emphy, and their polarization is known to be important for the lymphatic clearance. So I just want to show you some of the imaging experiments where we see some very striking results. So this is transmission electron microscopy. We use gold particles to label aquaporin-4. So before stimulation, you can see they are in the astrocyte emphy, but they are diffusely distributed. But after the stimulation, you can see they show this extreme polarization at the apical membrane. Okay, so this stimulation really induced aquaporin-4 polarization. This is a super resolution microscopy also show a similar results. So finally, I'm going to show you this result. The whole point that we do this experiment, which is to see whether this process is important for amyloid clearance. So, so we look at prefrontal cortical amyloid plaques 1 hour after stimulation, and with animals treated with saline or TGN-020, which is an aquaporin-4 inhibitor to block lymphatic clearance. So here we show with stimulation, we saw reduction of amyloid, but when we inhibit the lymphatic clearance, this clearance is ameliorated. So this experiment showed that indeed genus-induced amyloid clearance is mediated by the lymphatic clearance. So I show you a lot of things so far that 40 hertz stimulation can increased CSF influx, alpha-4 polarization, arterial pulsations, amyloid clearance, and so on. But can I find a single molecular event that can tie all of these different observations together? So I want to show you this a piece of biology in terms of synaptic release of neurotransmitters or neuropeptides. So neurotransmitters are stored and released by synaptic vesicles, and their release can be triggered by neuronal firing, but it really doesn't matter what kind of neuronal firing, low frequency, high frequency, it really doesn't matter. But the neuropeptides are known to be stored and released by the so-called dense core vesicles. I think you heard from Isaac presentation earlier. And these vesicles release are known to require high frequency bursting neural activities. And indeed, in the literature, it has been shown that for VIP release, 40 to 50 hertz neural activity is ideal. So, so then another thing also is that the vascular endothelial cells, as well as the astrocyte and feet, they all express high levels of neuropeptide receptors, VIP receptors, MPY receptors, and so on. So based on all of this, we wanted to test a hypothesis. We think that the high frequency neural activity trigger the VIP neurons to release VIP neuropeptides, and the VIP neuropeptides in turn binds to the receptors and trigger this cascade of downstream events to promote lymphatic clearance. So, so we perform a chemogenetic experiment to silence these VIP neurons. So we use VIP cremase to cross with the 5-FAD, and then we use the inhibitory DREAD receptor, and we introduce it by AAV, and this receptors is specifically expressed in the VIP neurons, and then we can use the designer drug to silence these VIP neurons, and we were able to show that the increased basal motion by 40 hertz now is completely blocked when VIP neuronal activity is silenced. And similarly, genus-induced amyloid reduction was also attenuated when we silence the VIP neurons. So I told you a lot of things. I just want to use this simple movie to summarize what I told you so far. I show you that sensory stimulation can lead to network activity. This leads to VIP release. I don't know, this movie is very choppy. But anyway, VIP binds to the receptors on the blood vessels and astrocyte emphy, and activate polarize alpha-4 and basal motion, increase CSF influx, and leading to amyloid clearance. So in the last couple of minutes, I just want to tell you that just when you think that you solve all the problem, it's usually not the case. So, so it turned out that VIP is not the only answer to the regulation. So, so this observation was made by Nick Li-Huei, which is going to be the first year graduate student in the lab. So he did a whole brain CFAS labeling to see brain regions that are activated by audiovisual stimulation. So this is just, so the brain was cleared using clarity switch process, and then we use light sheet microscopy to look at the whole brain CFAS distribution. And of course, a lot of computational analysis, segmentation of brain regions according to Allen Reference Brain Atlas. And after all of this work, we found that one of the brain regions showed the highest level of CFAS positive cells compared to non-stimulated animals is medial septum nucleus. So this is part of the basal forebrain housing the cholinergic neurons. So, multiple regions in the basal forebrain, such as the medial septum, show increased CFAS positive cells, as well as the vertical diagonal band of BRCA also show increased CFAS positive cells. So these neurons can project to the hippocampus and multiple cortical regions. And the CFAS, increased CFAS positive neurons is associated with increased acetylcholine signal. So here we got the grab sensor ACH 3.0 from Yulong Li at Beijing University. And we use fiber photometry to look at acetylcholine signaling. And you can see with stimulation, you see a peak of cholinergic signaling. And this is blocked when we treated the animals with scopolamine, which is a sort of cholinergic inhibitor. So, this then we realized that cholinergic signaling is actually necessary for genus-induced vasomotion, because here when we block cholinergic activity using these inhibitors, this vasomotion is inhibited. And most importantly, genus-induced amyloid reduction is completely ameliorated when we block the cholinergic signal. So, so just to cut a long story short, what we found is that cholinergic activation and innervation into the brain facilitates VIP release. So, so we realized that when we block cholinergic signal, then the increased VIP release is also completely blocked. So we use several different techniques to demonstrate that cholinergic signaling is necessary for VIP release. So, so basically, so far we identify a cascade of events, including involving multiple different neuromodulatory systems, including neurotransmitter and neuropeptides, underlying sensory stimulation induced neuroprotection. And today, I think since our initial publication, there are a lot of human clinical trials are ongoing based on the 40 hertz stimulation. There are more than 100 clinical trials ongoing targeting many different diseases. And I think it will be very exciting to see the results. And here, I just want to thank all of the members of my lab for contributing to this work. And particularly, I want to thank my collaborators at Boyden, Annabelle Singer, and Emery Brown, as well as the funding agencies. Thank you very much for your attention.
All right, questions?
Okay, yeah, that was great, fantastic, wonderful. I was wondering if you could talk about if you know anything about sleep. So so obviously the glymphatic clearance and many of the other mechanisms you discussed are going to be highly dependent on vigilance state, and gamma oscillation will also change as a function of the what vigilance state you're in, right? So
yeah, so I think there are two sub-questions. One is whether gamma oscillations has an impact on sleep. And in animals, we haven't really looked into sleep, but from a lot of the human studies now, people have shown that this treatment improves sleep and also improves circadian behavior. But how does gamma lead to that? I have no idea. But in terms of the mechanism of genus-induced gynvatic currents, whether that has anything to do with the increased gynvatic currents during sleep. I think that is really a very interesting question. Mike and Nadegar and I have talked about it a lot, so I think it will be interesting to find out.
Yeah, that was great. I was going to ask the same question, but maybe another question is, what's this last part you showed with the basal for, like, why are certain neurons being more sensitive to the gamma oscillations?
Yeah, that is also fascinating. We definitely see certain brain regions show much more robust response than some other regions. We obtain very similar results, actually using in animal model, using whole brain CFOS, and in even humans. So now we have the MEG imaging results, and we found that the response in humans and mice brain in terms of which brain regions particularly respond well to the simulation, they basically completely overlap. So we think that there must be a neurocircuitry mechanism in terms of how the signals are propagated through a particular pathway. So in the mouse model, we are now trying to, using optogenetics and chemogenetics to manipulate those regions and figure out what's going on.
All right. Yeah.
Thank you. A quick question is, I'm curious about how this VIP interneurons coordinate this all whole brain cleaning system, the lymphatic system.
Yeah, so VIP is known to be vasoactive intestinal peptide. So it has a very strong effects on the blood vessel motility. So, I think that with increased VIP release binding to the endothelial cells promotes vasomotion. And VIP also binds to the astrocyte and feed to promote alcoholin 4 polarization.
Yeah, and the lymphatic also play an important role during sleep. So do you think this VIP interneuron also, this mechanism also they play the same?
That's precisely what I told Jonathan. We don't know yet because I think during sleep, people haven't looked at whether VIP signal is increased. So I think it will be interesting to look at.
Okay, thank you.
Thank you. Thank you. Okay.
Thank you. Thanks to all the speakers. Our final event for today is okay. Our final event for today is the ceremony for this year's HCLS Distinguished Research Award. This year, we received a large number of high-quality applications. Each candidate was carefully evaluated by 13 reviewers based on innovation, significance, and quality of the work. 12 of them were selected for this award. Congratulations. Now I'll announce the 12 awardees who have made outstanding scientific contributions in the past year. We are honored to have Dr. Li-Huei Tsai here to present the awards to the recipients. Dr. Tsai, please take a photo with each of them after presenting the award. Now I'll call the names. When I call you, please come to the stage. Dingpeng Zhang. Siwei Chen. 庆庆奖。